Good afternoon, everyone. Thank you for the kind introduction, and thank you for sticking around for the last talk in the session. Hopefully, it will be light and entertaining, keeping you tuned in. As introduced, I'm a PhD candidate from Hanover Medical School, and it's my pleasure to share with you today the latest insights employing our technology that can generate iPS-derived macrophages, which we believe would be a very valuable tool to be employed in the MPS screening modules for immunotherapeutics. To understand and appreciate the value of incorporating human macrophages to MPS screening modules, we first need to study and learn the unique criteria of macrophages. As you all know, human macrophages are the innate immune cells which are responsible for maintaining the body homeostasis and defending the body against foreign pathogens. Also, macrophages are known for a multitude of unique hallmarks and criteria. In the recent years, macrophages have become very popular for their diverse ontogeny, which therefore can also extend to the diverse functionality. The traditional belief that all the human macrophages stem from a single source going back to the hematopoietic stem cell in the bone marrow or the circulating blood monocytes have been overruled by the latest data, which showed also that human macrophages are actually seeding organs before birth, having embryonic origins, and having the capacity to self-replicate themselves and maintain themselves in the human body throughout the lifetime of the human being. And this special population of macrophages referred to the tissue-resident macrophages. Indeed, having the macrophages distributed across different organs and tissues in the human body add to their diverse functionality, where macrophages can gain specialized set of functions, allowing them to maintain the health of the tissue they reside in. For example, cardiac macrophages will be specialized in maintaining the health of the blood vessels and facilitating the electrical conduction of the cardiomyocytes, whereas the alveolar macrophages in the lungs will be specialized in clearing the excess mucus to maintain the lung homeostasis. Not only can macrophages acquire diverse functionality based on their anatomical location in the human body, but also macrophages are known for their diverse activation status. Macrophages can acquire either a pro-inflammatory phenotype or an anti-inflammatory phenotype based on the environmental cues and the need of the human body, what is also known as the macrophage plasticity. Of course, this plasticity has to be finely balanced and tuned, otherwise it will result in the disease onset and progression. And given the previous information that I shared with you concerning the universal distribution of the macrophages in the human body, you can therefore appreciate and understand that disturbing this balance would really result in an array of multiple diseases across different areas of the body where macrophages can reside. And therefore, this also highlights the need to screen for macrophage targeting therapeutics and the role of macrophage targeting therapeutics in resolving human diseases. For instance, in the case of myocardial infarction, the cardiac macrophages are usually skewed toward the pro-inflammatory phenotype, which is actually worsening the disease progression because in that case, the patient is already suffering from chronic inflammation and fibrosis. Luckily, though, we can also exploit macrophage plasticity therapeutically by administering different macrophage modulators, which can vary from small molecules, cytokines, chemokines, monoclonal antibody, which can therefore rewire the macrophage phenotype from a pro-inflammatory to an anti-inflammatory phenotype, which would be very helpful in this case, resolving the disease progression and mediating the angiogenic needed effects. This is only one example where macrophages and macrophage-targeted therapy can immensely help in enhancing the disease phenotype. And therefore, we're proposing, for instance, that macrophages can be added to the sophisticated MPS screening modules in an isogenic complementary fashion, where, for example, here, an MPS that is established to culture a cardiac myocyte coming from a patient can be complemented with the patient isogenic cardiac macrophages, and then this system can be used to screen for different macrophage targeting therapeutics, which can be coupled to automated readouts. However, incorporating human macrophages in such sophisticated and robust screening modules might face several hurdles. Currently, the human macrophage resources are limited to two main sources, either the isolation from the peripheral blood or from using macrophage-like cell lines. And as previously mentioned, those two sources might not be very ideal, because while using the monocytes from the peripheral blood might be representing the physiological status, but it is limited to the donor variability, donor availability, the limited isolation yields, and the handling artifacts. On the other hand, using a macrophage-like cell line might solve the limited isolation yields, but it's also coming with the cancerous origin, the mutations, and the high variability upon long-term passage. So we can appreciate and also learn that still adequate and standardized sources of human macrophages are pretty much needed to complement sophisticated screening systems like the MPS. And for that purpose, I would like to introduce the induced pluripotent stem cells as a regenerative tool for continuous macrophage production. As you all know, this Nobel Prize awarded technology can allow us to recruit any somatic cell from any donor and further reprogram that cell to the pluripotent status, and then we can educate that cell to differentiate into any cell type of the 3 germ layer lineages, including the blood and including the human macrophages. And given the indefinite self-renewal capacity and the unlimited differentiation potential of the iPS, it can really make an attractive platform for continuous standardized supply of human macrophages. In our lab back in Hanover, we have been specializing for the last 10 years in establishing robust protocols, which are also nowadays GMP compliant, that can drive the generation in a continuous fashion of macrophages from human iPSCs. Briefly, our protocol is composed of 4 main stages. It starts with culturing the induced pluripotent stem cell, followed by expanding those cells to form what is so-called as the heminoids, or the embryonic bodies, those 3-dimensional structures. And then we educate those 3-dimensional structures to commit to the mesoderm lineage by adding special cytokines or specific cytokines. And thereafter, once those 3-dimensional structures have matured, all what we need to do is to seed them in any culture vessel, and then they will serve as factories for continuous production of human macrophages. And all what I need to do usually is go to my lab once weekly and then harvest the macrophages that are shedded to the supernatant in a continuous fashion. And as also tested by my own hands, this protocol can last at least for 6 months continuously from the same culture that you started. One of the many benefits of using iPS-derived immune cells in drug screening is, of course, as pointed out before, the scalability and the standardization. Not only in our labs we have established our protocols for small-scale production of human iPS macrophages, but also we have spent time and effort to transition this protocol to different production scales of different sizes. We have transitioned the adherent-based culture to a suspension-based culture that can support the culture of the iPS in different sizes of bioreactors, going from an intermediate scale of a 50 ml culture volume all the way to industry-compliant bioreactors that can go up to 5 liters. And having the scalable platform is coming with, of course, the advantages of guaranteeing a constant quality, high purity, and industrial quantities of the iPS-derived macrophages that you get from every harvest. And therefore, this scalable platform can really pave the path of incorporating human macrophages into diverse bioassays, including, of course, drug screening. To further exemplify the scalable production of the human iPS macrophages, I here demonstrate a video of a bioreactor that is running or was running in our labs in Hanover. And this bioreactor over here is having an iPS differentiation to macrophages. And the snowflakes that you see in constant rotations, those white snowflakes, are the beautiful heminoids or regenerative bodies that I referred to previously in our protocol. And they are constantly shedding the macrophages to the supernatant, and that's why the supernatant here is looking yellowish. At that point in time, all what we need to do is to stop the bioreactor, collect the supernatant, and therefore harvest the macrophages, and then we just need to refresh the heminoids with fresh medium and fresh cytokines to keep them continuously producing macrophages on a weekly basis, or for as long as you have the money to support this bioreactor. In this case, of course, we traced the bioreactor to 7 weeks, and we had an industrial production of the macrophages going to 10 to the power of 8 of iPS-derived macrophages from a 1-liter bioreactor tank. Not only is this process of a continuous and scalable nature, but also it's highly standardized, what we learned to be a very, very important criteria for cell-based assays. From the weekly harvests, you can see we were monitoring the quality of the iPS-derived macrophages, and as by the microscopic and the phenotypic analysis, the macrophages coming from different harvests were always showing a typical macrophage morphology and phenotype. So therefore, those illustrated features concerning the scalable and standardized production of iMonoMac or iPS-derived macrophages can really facilitate their incorporation into a sophisticated MPS screening module for immunomodulatory agents, where we're proposing to combine our scalable production techniques of the iPS-derived macrophages together with our capacities to genetically engineer iPSCs to produce what is so-called as the sensor or the reporter iMonoMac. Such a sensor cell can be easily traced in a microfluidic chip, providing more or less a high-throughput screening module. And the overall vision is not only to have an iMonoMac chip, but to rather incorporate that chip into a multicellular screening system where we can combine the iPS-derived macrophages in an isogenic fashion with other iPS-derived organs in a screening chip. To take a first step towards this vision, we first started by establishing the reporter iPS-derived macrophage, or the sensor iMonoMac, which is harboring an NF-kappa B pro-inflammatory transcription factor controlling the expression of the reporter. And this cell can thereafter be a very valuable tool to screen the different immune modulatory drugs or pro-inflammatory agents. We have achieved that by transducing an iPS cell line with a third-generation lentiviral vector that is SYN inactivated. And after establishing the cell line, we further differentiated it to give rise to the NF-kappa B luciferase reporter iMonoMacs. And then those reporter iMonoMacs can be, of course, used in different setups to screen for the activity of a pro-inflammatory agent or an immune modulatory drug based on the expression of the luciferase gene. We have confirmed the successful production of those reporter iMonoMacs as they again show the typical macrophage morphology and phenotype. And on a functional level, when we have incubated the reporter iMonoMacs with an array of different inflammatory agents, including, of course, the standard LPS treatment, we could nicely see a dose-dependent response where the LPS concentrations were sensitizing the luciferase reporter inside the macrophage, and we can thereafter measure the luminescence signal with a plate reader. Also, what is noteworthy to mention here that this assay was far more superior and sensitive compared to the traditional cytokine measurement that is usually used to monitor the activation of macrophages with inflammatory agents using an ELISA. Because an IL-6 ELISA have failed, and sorry, here there's a typo, this should be a different graph. This should show you an ELISA graph where the IL-6 secretion have failed to pick up the small concentrations of the LPS. If you didn't catch it here, you can catch it in my upcoming publication. Moving on to the second objective. So not only did we want to provide the reporter tool to screen for the individual drug entities, but also we wanted to move a step closer toward a multicellular screening system. And given the high potential of macrophage in cancer therapy, we like to develop a GFP constitutively active iMonoMac cell, which can be co-cultured with different cancer cells, which are usually expressing the M-cherry fluorescence. And then this co-culture setup can be used to screen different immunotherapeutics, including the phagocytosis checkpoint blockers. Indeed, targeting macrophage phagocytosis has received increased attention for cancer therapy because, as you know, usually cancer cells try to evade immune recognition by several mechanisms, among which is the overexpression of the phagocytosis checkpoints like the CD47. And usually the CD47 is interacting with the core receptor on the surface of the macrophage, which is the ZIP-alpha. And once this interaction happens, the phagocytosis of the cancer cell by the macrophage is blocked. And therefore, upon the recognition of this pathway, they were considered as highly valuable drug targets, and many of the monoclonal antibodies were in clinical evaluation to inhibit this pathway and therefore activate the macrophage phagocytosis, which resulted in magrolimab to be in phase 3 clinical trials. And here again, you can appreciate that macrophages are really attractive drug targets in the context this time of oncology. And therefore, having robust screening modules for macrophage targeting therapeutics is highly needed. And again, as I previously mentioned, we have established this time the GFP constitutively expressing IPS layer and differentiated that into the IPS-derived macrophages, which are constitutively expressing the GFP. They were showing the typical macrophage phenotype and morphology, and also, of course, to use this as a tool to screen for phagocytosis checkpoints, we had to make sure that the imonomac can express the phagocytosis checkpoint just like the primary monocytes, and indeed they are positive for the ZIP-alpha expression. And thereafter, we have established different co-culture systems of the GFP imonomacs with different cancer cells, including non-small cell lung cancer, which I'm not showing here, and also the Raji-Burkitt lymphoma as an example of a liquid tumor. And as you see, upon the addition of the phagocytosis checkpoint blocker, we can trace the increased phagocytic events both by the microscopic evaluation and also by flow cytometry. We have increased phagocytosis when the monoclonal antibody is added, but not when the control antibody is added, which is showing the success of the screening system. Lastly, taking things again a step further to be incorporated easier to the sophisticated MPS screening module, we also have provided another readout which combined the previous readouts of the flow cytometry and the fluorescent microscopy using an image-based flow cytometry, which is more of a high throughput nature as it can screen 2,000 events per second, and it can also complement nicely the fluidic system inside the microfluidic chip. And again, here you can see when we measured the phagocytic events, upon the addition of the monoclonal antibody, we have successful phagocytosis, whereas we don't see a similar trend upon the addition of the isotype control. In summary, what we are presenting you here is a very valuable cell type that can nicely be incorporated into the sophisticated systems of the microfluidic chips. Alongside to our technology of the scalable production and the genetic engineering, we can really provide a high throughput scalable system to screen for immunotherapeutics in general and macrophage targeting therapeutics in specific, which would be of very great value for the different human diseases. And at the end, I would like to acknowledge the help and effort that I always get from my colleagues back in the Lachman lab in Hanover Medical School. And thank you a lot for listening, and I'm happy to take your questions.
Thank you again. Nice talk. Some pretty snowflakes. First question.
Thank you for this nice presentation. I was just wondering, there is a paper 2019 published by Valia Orevas lab with a protocol already describing how to generate monocytes and macrophages. Where do you see the advantage of your protocol over this more or less established protocol? And the second question is, how would you describe the phenotype of the macrophages you're generating? Are these different monocyte-derived macrophages, or do they have any similarities to tissue-resident macrophages you were mentioning at the beginning?
Yeah. Thank you for the very nice questions. First of all, concerning the advantages of our protocols, you're right. Currently, there are many labs which are replicating or reproducing the IPS-derived macrophages using different protocols, but I can be very confident in saying that we're among the elite people in that field, because the very first paper that was published by my PI dates back to 2015 in Stem Cell Report. So we have a long-standing experience in that field, and now we also have, I'm not quite familiar with the paper that you mentioned, which protocol are they following, but concerning our protocols, we have transitioned to a completely GMP-compliant protocol, feeder-free protocol, and it requires the minimal supplementation of cytokines, which is usually a problem when it comes to kind of having those sophisticated protocols running for high purity of the macrophages. You tend to kind of nourish the protocol with a lot of cytokines. We only use IL-3 and MCSF for our protocol, which is an added advantage, at least also what we highlighted in our patent. And the scalability of the protocol is also a great advantage, because as well, since 2018, we have published our first paper concerning adapting our protocol in bioreactors and Nature Communication. So scalability, minimal cytokine supply, and The GMP compliant version that we currently have is also an advantage for the running protocols. Those are the three main pillars that come to my head at the moment. I'm sure I can come up with more. Yeah, and concerning the second question for the similarities and differences to the primary monocytes and to the tissue-resident macrophages, that's a great point. And we actually have a PhD thesis dedicated to study in detail the ontogeny of our iPS-derived macrophages using single-cell RNA sequencing and so on and so forth. But the insights that I can share with you is that we know, at least also what is shared from other publications, that iPS-derived macrophages are usually lacking the HLA-DR expression, which is a marker of naive or primitive iPS macrophage compared to the adult human monocyte. This, of course, could be an advantage or a disadvantage based on your screening or specific application. But having a naive origin of the iPS-derived macrophage therefore supports its capacity to mimic more the tissue-resident macrophage compared to the primary monocyte. And indeed, we have also other projects where we are differentiating the monocyte to alveolar macrophage, the iPS-derived macrophages to alveolar macrophages, and this works quite nicely. We're employing them in different culture setups like ALI and so on and so forth. So they are more of resembling a naive primitive phenotype, and they can differentiate nicely to a tissue-resident macrophage. And they are, what we compared also against the primary monocytes, that they are more sensitive and they are of a higher pro-inflammatory nature compared to the primary monocytes in our infectious studies. And this information is all also published, and I'll be happy to share it with you.
Thanks. Any more questions?
Can I ask if these are commercially available, and also can I ask of the donors, do you know the MHC type, and what sort of diversity have you got in the donor sets?
So that's a great question. We have started first by generating our own in-house cell lines. We had only a limited pool of cell lines, like 3 cell lines, and the MHC was class 2, I think. I'm not sure about the MHC typing, I have to check. But now we also are doing the same kind of validation studies on other cell lines that we're getting from collaborators and from commercial sources to kind of further validate our protocol and move a step forward to the clinics. So yeah, this is concerning your question. Yeah.
Any more questions?
Thank you for the talk. Maybe it's a naive question, but can you explain better how the iMonoMac chip works and how do you take the output of the data?
The iMonoMac chip is still in the planning phase, so we're promoting our tool to be used in an MPS screening module, but unfortunately we don't have the expertise in that area. We're collaborating currently with a lab in Mainz, but we're still running some issues concerning the funding to have the project running. So we have a prototype, but still we haven't generated any data from that module, let's say. But I'm rather kind of presenting the tool itself, which can go there, hopefully, with your help. So yeah.
Thank you. Maybe one last question if it's quick. Okay, let's thank our last speaker. Thanks for coming, and thank you to our speakers for keeping everything moving along.